Hello, welcome to my video, and we're going to talk about cellular respiration in this video. So you are going to want to find the page that has a diagram that looks like this. Draw the basics of what I have on the diagram. So I've labeled the diagram. We have the cell membrane. Okay, this whole space is the cytoplasm. I've labeled it up here. And then I just wanted to draw one organelle, the mitochondria, in my cell to show, because we're just doing cellular respiration, so just the mitochondria is being used. There obviously would be a nucleus, there would be a whole bunch of other stuff in this cell. I just wanted to make sure that I wasn't overcomplicating the diagram. So we talked about this in class, but this is the mitochondria. We have parts of the mitochondria, like the matrix, that's the space in the middle. The cristae is kind of the squiggly line. And then between the cristae and the outer membrane of the mitochondria is what we call the inner membrane space. So we have outlined the different steps. You have glycolysis, the intermediate step, and the Krebs cycle, and then the electron transport chain. So we're going to go through and talk about all of these steps. But first we're going to start by filling in the equation. So I drew it just slightly below, just because, as you can see, my little diagram is really, really small. So we start with glucose and oxygen. And then after the reaction occurs, we end up with carbon dioxide, water, and of course ATP. So in order to do this equation, we start by taking glucose in our step glycolysis. Remember, that means literally glucose and to break. So I have 6 carbons in my glucose molecule. I'm going to take those and break them into 2 3-carbon molecules that I call pyruvate. So once I take my glucose, I make pyruvate. Doing that process, okay, releases energy, and I'm going to store the energy as NADH. I'm going to end up with 2 ATP. So remember that there's actually 4 ATP that go into this process, glycolysis. 2 is used up to break open the glucose, so I have 2 left over. That's where this number 2 is coming from. So my 2 pyruvate that I have broken are still a little bit too big in order to fit in the mitochondria. So I'm going to break them up even more into a 2-carbon compound and a 1-carbon compound. Okay, the 1-carbon compound will attach to some oxygens, and we can get rid of them because we don't need CO2 in our cell. Eventually, it'll be sent through our blood to our lungs, and we'll breathe that out. This 2-carbon compound is officially the right size to fit into our mitochondria. It's called acetyl-CoA, and it can start going into the Krebs cycle. But this process of creating acetyl-CoA actually also produces 2 NADHs. So in total, between this 2 and this 2, we have 4 NADHs. So those are going to be used later in the electron transport chain, but in the meantime, we have this acetyl-CoA that's going to be going into the Krebs cycle, my 2-carbon compound, acetyl-CoA. In my Krebs cycle, it's a process where a bunch of stuff is happening. Okay, so basically kind of similar to the Calvin cycle in photosynthesis, where you're putting things together, breaking them apart, putting them together. We eventually end up making carbon dioxide, which again is going to be released from the cell as waste because we don't need it, put into our blood, eventually ends up at our lungs, and we breathe it out. Okay, we also have another energy storage molecule, one that we haven't talked about yet. It's called FADH2. So again, energy storage molecules are just in charge of basically storing energy in the bonds between the elements. So we're going to use that to power the electron transport chain. We have 2 FADH2s that we create. We also have 6 more NADHs. So in total, right now, we have 10 NADHs, 4 plus 6, and 2 FADH2s that are going to be the energy source for the electron transport chain. Now, on the cristae, I've zoomed in on it, and on this cristae membrane, you have these membrane proteins. So you have ATP synthase and some other membrane proteins that are going to open and close and allow hydrogen ions into the inner membrane space. So this space right here is going to fill with hydrogen ions, build up that concentration gradient that we need for the electron transport chain to occur. So before the electron transport chain occurs, we need electrons in order to do this process. So what we're going to do is we're going to take some water that's just kind of chilling in the cytoplasm, and we're going to break it down to steal some electrons from that molecule, from this reaction. So when we break water down, we end up with hydrogen ions, we end up with oxygen. So we're going to take the electron, and it's going to be energized by NADH, and the FADH2 is going to energize it, pump it up, get it ready to go. So once it's energized, it's going to shoot across the membrane protein. It's going to allow the membrane protein to open. Every time the membrane protein opens, it allows a hydrogen ion to go in the inner membrane space. Okay, this hydrogen ion could be from this process right here where we're breaking down water. Okay, or maybe you have some hydrogen ions just kind of floating around in the cell, and they'll be going to the opposite side once the channel opens. So once it shoots across, it's again going to be re-energized by the NADH or FADH2. It's going to shoot across again. We're going to allow another hydrogen ion into the membrane. And remember, building up that concentration gradient on the inside of the inner membrane space. They're uncomfortable. They don't want to be there. They're going to push themselves through this ATP synthase molecule, which is going to cause the ATP synthase molecule to spin. Once it spins every single time, it's going to smack together an ADP, which is adenosine diphosphate, 2 phosphates, with another phosphate to make ATP. So this is a little turbine spinning and spinning and spinning every time a hydrogen ion spins through. And we're going to create a whole bunch of ATP, okay, using that concentration gradient that we've built up. That's what's happening here. Every time it goes through, it's going to spin. We're going to create a whole bunch of ATP, which is the whole point of this process. So because we have actually a whole bunch of energy, so 6 NADHs, 2 FADH2s, we're going to create in total, at the end of the electron transport chain, about 36 ATP. So in total, I had 36 ATP, you can't see it right now, and then I had 2 ATP over here. So in total, after I'm done all of cellular respiration, I finish with 38 ATP. Okay, now we have some other products that we're left over with. So we have some electrons, we have some hydrogen ions. So we want to do something with these things. We don't want to just leave them there, because eventually they'll build up and build up and build up and cause problems. So we don't want them to just hang out and do nothing. So what's going to happen is we have this oxygen that's just hanging out, okay, waiting for the electrons to come. It's called an electron. What do I call it? The electron acceptor, sorry. So electrons and hydrogen ions and oxygen is going to be combined to create more water at the very end. So that's what I'm showing here. So oxygen is my electron acceptor. It's going to be combined with hydrogen ions that are built up and the oxygen that we had, and then the electron. It's going to make a whole bunch of water, which again, we don't need. It's a waste product. So it's going to just basically be breathed out in our air that we breathe out. Okay, that's why when I go against the glass, I draw a little smiley face. It's because you're actually breathing out water as well. So that is the end of cellular respiration. Okay, it seems complicated now that I'm looking at the diagram altogether, but the whole point of cellular respiration is to make 38 ATP, which is a whole bunch of energy, so we can use it for cellular processes, muscle contractions, okay, different processes like getting rid of waste from our cell, allowing nutrients into our cell. Okay, there's a bunch of different processes that need this energy, and without this process, this regular cellular respiration, which I would call aerobic because oxygen is present here, we would not be able to move. Or we could move very inefficiently if we didn't have oxygen present. So cellular respiration is basically keeping you alive every moment of every single day. So that is exciting. Now, make sure you guys draw this out a few times. We will have a drawing quiz about this tomorrow. If you have any questions, please let me know. But if not, have a wonderful evening.